So it's my pleasure to introduce Megan Cerny. So Megan's a double major in biology and chemistry, and she'll be getting a minor in business as well. And today she's going to talk about some research, some biological research she's done with Dr. Matthew Stephenson. And so Dr. Stephenson's currently in Austin, Texas, and so I'm the on-campus advisor for Megan. So I'll let Megan take it away and talk about some of the research she's done with Dr. Stephenson. So let's give Megan a hand.
Thank you. So as Dr. Kleinschmidt mentioned, my name is Megan Cerny, and today I will be discussing the immunological cost of autotomy in pinhead crickets. So to begin this presentation, it's not working. First, I'd like to give an outline of what we'll be discussing. So first, I'd give some background information on what autotomy is, and then we'll look at the immune system in pinhead crickets and some different energy allocations. Then we'll look at the methods that I use to conduct this research. We'll look at my results, and finally we'll look at a conclusion and some of the future implications that can improve this research. So autotomy is the voluntary abandonment of an appendage that many invertebrates do, and they usually do this as a defense mechanism. So as I mentioned, many invertebrates can do this. So as you can see, this spider here only has 6 legs. He should have 8. And this cricket up here, he's missing his right hind limb. So as I mentioned, this is a voluntary abandonment of that appendage as a defense mechanism. So let's say that a predator has grabbed either a leg of a spider or the leg of the cricket. That invertebrate can voluntarily abandon that limb to allow it to escape that predator. We also see this happen when an invertebrate has been stung by a wasp and there's venom within that appendage. They can voluntarily abandon that appendage to decrease the spread of that venom. So there's been a lot of research that looks at autotomy, and they've really been trying to investigate and identify a downfall after autotomy occurs. So they've looked at the difference between predator evading, prey capture, and daily mechanics of invertebrates that have undergone autotomy and those who have not. And they haven't really been able to identify a downfall after autotomy occurs. And so research now is moving from looking at more external movements and evading to more internal downfalls. So this brings us to energy allocations. Energy allocations fall within the life history theory. And this theory states that energy can be allocated to 3 main categories. The first is the growth category, the second is reproduction, and the third is survival. Now, where the energy is allocated really depends on that organism's body condition, environment, and age. So let's say an organism was just born. Most of its energy will be allocated into the growth category. And then let's say that same organism reaches sexual maturity. Energy will move from the growth category into the reproduction section, so that energy can be allocated to producing eggs and sperm, and also the energy that's needed to attract a mate. So research, or I guess life history theory classically had the immune system fall within the survival category. However, researchers now are really pushing that the immune system is the 4th category of energy allocation. And that's because the immune system is so large in the organism as a whole. So the immune system is every organism's way to fight off foreign entities. And they do this by fighting off that foreign entity after it's breached the skin or cuticle. And the cuticle is essentially the skin of invertebrates. So there's 2 different immunological response mechanisms. The first is the innate system, and the second is the adaptive system. The innate system is a very general approach to fight off a wide variety of pathogens, and the adaptive system is more of a specific approach to fight off a very specific pathogen. So humans have the adaptive system by using antibodies against antigens, and crickets solely have the innate system, so they don't have the adaptive system at all. So in this presentation, I'm mainly going to focus on the innate system. So to show you what the innate pathway looks like within crickets, let's say that this circle denotes the cricket as a whole. And let's say that a foreign entity has breached that cuticle of the cricket. So now this foreign entity is found within the organism itself. Within the hemolymph, which is essentially the blood of crickets, we find these recognition proteins, and they're very abundant within the hemolymph. So these recognition proteins have the ability to recognize foreign entity versus self, and once they do this, they will undergo a conformational change, and this will elicit a chemical response to the rest of the cricket, saying that there has been a foreign entity present and that it's under attack, and this will recruit more proteins to help fight off this infection. So one of the main cells that receives this signal is a hemocyte, and hemocytes are essentially the blood cells of invertebrates. And once these hemocytes receive this signal, they'll start changing their internal chemical pathways to allow them to be ready to fight off that foreign entity. So let's take a quick look at what's going on within the hemocyte themselves. Hemocytes have an enzyme called prophenol oxidase, and when the hemocytes receives that signal, prophenol oxidase will start changing into phenol oxidase. Once this occurs, phenol oxidase will then undergo many different pathways and eventually produce melanin. Melanin is the hemocyte's main ammunition against foreign entities. So while this is occurring and those hemocytes are being recruited to that site of infection, the hemocytes will start encapsulating that foreign entity. And this really deprives that foreign entity of nutrients and decreases the likeliness that that foreign entity is going to spread throughout the body. Then the hemocytes will start depositing that melanin, which was that main ammunition towards the foreign entity, and they'll start depositing it to further encapsulate that foreign entity. Once this occurs, melanin will start producing reactive oxygen species. And reactive oxygen species are very damaging to cellular wall components as well as proteins. And so these reactive oxygen species have the ability to completely disintegrate that foreign entity into smaller pieces that are no longer able to infect the organism as a whole. The problem with these reactive oxygen species is that because they are so reactive, they can actually escape that encapsulation event by melanin, and they can start damaging the cellular components at that site of infection. However, another enzyme that was recruited to that site of infection by that chemical signal that was sent by the recognition proteins is superoxide dismutase. And superoxide dismutase has the ability to neutralize those reactive oxygen species to hydrogen peroxide, and then the hemolymph can further reduce that hydrogen peroxide to water so that it's no longer damaging to the organism as a whole. So now that we know what's going on within the cricket when a foreign entity is present, different energy allocations, and what autonomy is, this brings us to the main focus of my research. So in this research, we decided to look at, once autonomy occurs, is energy being allocated to the immune system in pinhead crickets? So now on to the methods. The very first step of our methods was to obtain pinhead crickets, and then we separated them out by sex and disformities. So this arrow here points out the ovipositor in female crickets, and this ovipositor allows females to deposit eggs within the ground. So we selected only male crickets, so we selected the crickets that lacked that ovipositor. And this cricket here is missing its left hind limb, and we wanted to actively autotomize the cricket's legs. So we selected against crickets that have already undergone autotomy. Once we had enough male crickets without disformities, we placed them into 3 different groups. We had the control group, a 2-hour autotomy group, and a 4-hour autotomy group. Now I do want to mention that at the beginning of this research, each group had 15 crickets within each category. However, during the hemolymph extraction, we had 9 fatalities in the control group, so their hemolymph was not able to be used for our data analysis. So once we had the male crickets in their specific groups, we started doing the autotomization. And in order to do this, we gently pinched the hind limb of the cricket, and they voluntarily removed that limb. So to show you what this looks like, you can see that the forceps are gently pinching on that spider's leg, and that he very voluntarily removes that limb. So once autotomy occurred, we introduced soil to that site of autotomization, and in order to do this, we gently picked up the crickets and gently rubbed that site of autotomy into the soil, and this induced foreign entities to be found within the organism itself. Once each cricket of each group were autonomized and had soil introduced, they were allowed to incubate at room temperature for their allocated time periods. And then And then we started extracting their hemolymph. And so we placed them in the anesthetic chambers to allow them to be placed under anesthesia so they didn't feel the extraction of their hemolymph. And in order to extract the hemolymph, this was by far the most difficult process of our methods. And this is also where we had our 9 fatalities. So at first we started using these push pin needles to penetrate the abdomen of the crickets. And the problem with this is that we really needed a uniform sphere of hemolymph to form on their abdomen so that we could get a large enough concentration of hemolymph. However, the push pin needles spread the hemolymph out on the abdomen of the crickets, and so we are not able to actually pick up any hemolymph. So then we moved to these hypodermic needles, which are a little bit sharper. So we thought having a smaller hole would allow that uniform sphere to form. However, as soon as we penetrated the abdomen of the crickets, that hemolymph shot straight into the column of this hypodermic needle. And then we were not able to get that hemolymph back out of the hypodermic needle. So essentially what we ended up doing after troubleshooting through 9 crickets, we melted parafilm and essentially clogged that hollow column of the hypodermic needle. And then we were able to penetrate the abdomen of the cricket and form that uniform sphere. And then we were able to get enough hemolymph to use in our data analysis. So once the hemolymph was extracted, we placed them in these microcentrifuge tubes with a phosphate buffer and glass beads. And we vortexed them for a few seconds to induce lysis of the hemocytes, so essentially breaking up the blood cells. And we did this because we wanted to look at specifically phenol oxidase, prophenol oxidase, which is found within the hemocytes themselves, so we had to break up the hemocytes. Once we did this and had enough hemolymph from each of the crickets, we placed them in these microtiter plates with the respected reagents of the assays that we are doing. And then we placed them in the absorbance spectrophotometer and did colorimetric assays. So what this does is if the protein of interest was present, a color change would occur because of the reagents. And then placing them in the absorbance spectrophotometer, light is shown through the wells, and the amount of light that's absorbed by each well is directly proportional to the color change that is occurring, which is also directly proportional to the amount of protein in each well. After we got our raw data from the absorbance spectrophotometer, we did statistical analysis to infer statistical differences between the 3 different groups. So now let's look at the results. This first graph shows total protein concentration. On the x-axis, we have the 3 different groups. We have the control group, the 2-hour group, and the 4-hour group. On the y-axis, we have total protein concentration in milligrams per milliliter. And the letters above each column represent statistically different groups. So what we were able to see within this graph is that the total protein concentration increased from the 2-hour group when compared to the control group, as well did the 4-hour group. However, when looking at the 2-hour autonomy group, the difference was, the increase in total protein concentration was statistically different when compared to the control group. However, the 4-hour autonomy group was not statistically different from the 2-hour group nor the control group. So then we looked at superoxide dismutase activity, and again on the x-axis we have the 3 groups. On the y-axis we have the percent SOD activity, and then the letters again represent statistically different groups. So what we were able to see with this graph is that the SOD activity did not change very dramatically between the 3 different groups, and they were not statistically different. So the 2-hour group was not statistically different than the control group, and neither was the 4-hour group to the control group. Then we looked at the prophenol oxidase activity, and on the x-axis again, we have the 3 different groups, and on the y-axis we have prophenol oxidase activity, and the letters again represent statistical different groups. And in this figure, we are really able to see that the 2-hour autonomy group had a very large increase of prophenol oxidase activity when compared to the control group and the 4-hour group, and it was statistically different from both. However, the 4-hour group did increase in prophenol oxidase a little bit, but it was not statistically different from the control group. So now you might be asking, what does this all mean? So within our results, we were able to suggest that the protein activity did change within the 3 different groups, and they changed as well in the different time periods. And we were able to suggest that the immune system was activated after autotomy occurred, and this is really by the difference in prophenol oxidase concentrations. So we saw that in the 2-hour time group, the prophenol oxidase activity dramatically increased, and it was statistically different than the control group. So this might suggest that one downfall to autotomy is that the energy must be allocated to the immune system to help fight off that foreign entity. Now, I do want to point out that, to my knowledge, this is the very first study that has shown a correlation to autotomy and the immune system, and we plan on publishing a paper into the Tri-Beta journal. So some future implications and improvements to this research. I feel it would be a very great idea to use a calibration curve so that we can definitively say that the amount of foreign entity present is directly correlated to the amount, the amount of foreign entity present is directly correlated to the amount of proteins being produced. And placing a standard concentration of foreign entity into each cricket so that there's no differences in between the crickets and their protein concentrations. In this study, we solely introduced soil, and that soil can have many different concentrations of foreign entity. And so I think it would decrease the variance in the time groups with the amount of protein being produced. And I also think it would be really interesting to look at shorter time frames, so looking at half-hour incubation times instead of 2 hours. And I think that this would give further insight of how fast that immune system is being activated. So with that, I thank the Porter Scholar Committee for providing funding and the resources to conduct this research. I thank Dr. Stephenson for being my advisor within this research and really helping me conduct it. I thank Dr. Kleinschmidt for stepping in as my advisor in the absence of Dr. Stephenson. And I thank Adams State University as a whole for allowing me access to the facilities as well as the instruments used within this research. There are my references. And with that, I'd be happy to take any questions.